Today we will talk about the actual technical details and some of the more important aspects of sequencing as it relates to SARS-CoV-2, and a lot of it is generally applicable in molecular diagnostics of SARS-CoV-2. By now, we should be quite familiar with the fact that there exists a reference genome for SARS-CoV-2 in GenBank. It's designated as the reference genome because it was specially curated, and it's standard for all important organisms to have a reference genome. If you go into the GenBank record for the sequence, you will see as the first reference this Nature paper in 2020 by a large number of authors where this sequence was originally generated. So what we can do next is go to that paper. So this was published in February of 2020 at the very beginning, I believe, of the month. They generated the sequence. So let's try to figure out by scanning some of the more salient parts of the paper about how they did this. The sentence in the abstract that pertains to this reads that metagenomic RNA sequencing, described in reference 4, of a sample of bronchoalveolar lavage fluid from the patient identified a new RNA virus strain from the family Coronaviridae, which is designated here WH human 1 coronavirus and is also being referred to as 2019 nCoV. So, and then there's some other stuff about phylogenetic analysis and characterization. So the key phrase here is metagenomic RNA sequencing. So it'll take us a little while to unpack what this all means, but the goal of the first part of my lecture is to give you, enough understanding that you can get a sense of how these types of sequencing studies are done. And in particular, it sort of brings up another broader question. So SARS-CoV-2 isn't the first. Zoonotic virus won't be the last. And if I would like you to recall what I mentioned in the first lecture, where one of the facts about viruses is that we know very little about them. We studied very diligently a small number of important human pathogens, but we haven't really surveyed what else is out there. And if a novel virus arises, like in this case, how do we discover new viral sequences? So we suspect it's a virus. We suspect it's not something we've seen before. So how do we discover what its sequence is like? And this technique of metagenomic RNA sequencing and metatranscriptomics is basically, you can think of this as a brute force approach, where you take an isolate from, in this case, a patient, and we'll discuss it in more detail. You try to sequence all of RNA that's in it. The reason you sequence RNA is you suspect that this is an RNA virus. It doesn't have to be, but you can do the same thing with DNA. It would be a standard, not metagenomics instead of metatranscriptomics. So you generate millions of partial DNA sequences with errors. And somewhere among those sequences, there are pieces of the viral genome that you're seeking to describe. So some of the questions that arise at this point is, how do we generate these millions of sequences? Supposing we're able to do this, how do we then identify new, meaning previously never seen organisms? So once we identify something new, how do we obtain high quality genomes and correct errors, which is presumably something that has to happen before it becomes a reference genome? And then once we've established the reference genome, the next question is, how do we scale this up to generate thousands upon thousands of genomes quickly to track the epidemic and understand what the virus is doing as it's spreading through the human population? It is not my aspiration to give you anything that resembles a comprehensive overview of sequencing. This is just very, very superficial, and I presume that a fair number of you have come across it in some capacity, especially if you're a biology major, because sequencing is one of the most fundamental techniques in modern biology and modern medicine. And roughly speaking, all sequencing approaches consist of two major steps. So the first thing, you have to prepare your sample. How you do this is very different from application to application. And a lot of people make careers quite successfully coming up with clever way to do sample preparation so you can use a standard sequencing technique to learn something new. But in this case, you would have, generally speaking, again, you would have to isolate or purify or amplify DNA in sufficient amounts. Typically, you will also take longer DNA molecules and chop them into smaller pieces for a variety of technical reasons. Then you will take these pieces and perform a number of technical steps to get the DNA in the sample ready for chemical or physical reactions that it is subjected to on the sequencer. A modern sequencer is a feat of engineering, nanotechnology, computer science, optics, semiconductor, fluidics. But what it really does, all of the modern sequencers, they perform millions of complex reactions in parallel. So you're basically sequencing millions of individual molecules in very, very small volumes or detection cells or nanopores. It depends on the actual technique that's being used to produce sequences. And each of these reactions will produce a detectable signal. It'll be very, very small. It's not something you can see with the naked eye, obviously, because you're doing something at a miniature or nanoscale. And the two types of signals that are produced in this context would be light or electrical current. They can be read out to determine, with some error, sometimes significant error, which of the four bases is in a particular position in the sequence. This is output as a large file, typically gigabases or gigabytes in size, with individual sequences and quality scores. And for example, you can see what these things look like. There is a database that is maintained by NCBI. It's called SRA, stands for Short Read Archive. It is basically it's a companion database to GenBank, which stores more traditional assembled genomes or sequences that represent full genes or full organisms, but more generally just generated with older techniques. A Short Read Archive is a special large volume database that stores these next generation large sequencing runs. So, and there, as of yesterday, there are probably more today, there were 52,358 SRA records for SARS-CoV-2. Obviously, it's also a very, very heavily studied organism. So I highlighted one here. So this was performed on an instrument called Illumina. The model of the instrument is NextSeq 500. And this particular run generated 200 million nucleotides. You can click on this and you will see a metadata of this run. So it's going to tell you it has an accession number, ERR 457, and so on. It'll tell you how many megabases there are. It'll tell you some general metrics about what the content, GC content of the reads in this sample is. So it's 38%, 38.5% when it was published. And there's also, there's some other information. So this was done using amplicon, which we'll discuss a little later, from viral RNA. And there are a couple of other key terms. So the length, the reference length, so you might recognize 29.9 kilobases as close to the length of the reference SARS-CoV-2 genome, and coverage is basically how many times each position was sequenced, so over 6,000. Will become, it's an important metric that I'll describe a little bit later. You can zoom in on specific reads, just go to a different tab. Obviously, it's not something you want to do by hand because there's 71,000 of them here, as you can see. But a typical read will look like this. It's going to have just it's a short string, so it doesn't represent the entire genome or even a substantial part of the genome. It's a small fragment of the genome. There's no particular rhyme or reason to why they're named this way. The instrument gives them a random name. So you can see these nucleotides, and underneath there is a quality score, which is, I'll describe again later, but the higher the score, the more confident you are that a particular base in the sample is correctly called, right? Remember, you're doing some fairly complicated chemistry, optics, and signal processing to call these bases. So some of them are going to be incorrectly called. So we'll come back to scores a little later. So this is the raw output, what a raw output from a meta-transcriptomic analysis like done in the Nature paper would look like. Millions upon millions of short reads, which are on their own obviously completely useless. You can't interpret them in any way other than to say I have some sequence data and it looks okay to me. All right, so let's talk a little bit more about, in about 15 to 20 minutes, about DNA sequencing. Again, I presume that a fair number of you have some exposure. By the way, I just realized I didn't have my video on, so sorry about that. Here it is. I guess it doesn't really matter that much. So most sequencing approaches will piggyback on natural DNA copying systems, typically polymerases or something along those lines, and modify this copying process to measure the nucleotide that's being incorporated. Right, so this is, and we're still figuring out novel ways to do this. You know, cheaper, faster, more portable, but that's pretty much all modern sequencing right there. We just, you know, hijack a biological process, we either stop it in some sense or, you know, spy on it to see what is actually being inserted as a DNA molecule is being copied or just being passed through some detection. And measurement is either done with light, in which case you rely on fluorescence, and that's detected either with lasers or really sensitive charge coupled device type systems, or ions, in which case it'll be detected through a change in charge, and it's done with semiconductors. So the third and fourth generation sequencing don't actually rely on copying. They do just called direct sequencing. So they try to read off DNA composition directly, but they also use naturally occurring systems, for example, nanopores for that purpose. I think it's worthwhile to go back to, you know, the time when I was a little kid, the late 1970s, to recall the genesis of modern sequencing. And there were sort of, at the time, there were two competing techniques for sequencing. So the Sanger, who's the first author of what became popularized in the 1980s and was sort of the dominant form of sequencing until the mid-2000s, called Sanger sequencing. And the actual details are not that important, but fundamentally what you do is that, like I said, you do, you modify a natural reaction of copying DNA. So in this case, the technical innovation was that you can take the nucleotides that normally would have been incorporated when the molecule is extended, double-stranded DNA, and you would modify it in two different ways. One, you would add it, you would add a detectable light emitter, so a fluorophore, which would be color-coded based on the nucleotide. So, you know, in this case, magenta, red, blue, and green. And you would also modify it chemically so that when this molecule was incorporated, the synthesis would stop. So with natural DNTPs, it would continue through, and here it would fall off. So you basically terminate the reaction and color it. So what this would create is that when you have DNA sample that you're trying to sequence, these positions, these labeled dideoxy molecules would get incorporated, stop the synthesis, and it was created a ladder of molecules of different length, right? So, you know, first position, second position, so on. That would be color-coded, and then you would sort them by size, for example, in later sequencing instruments using capillary gels, and you would have an optical detection system that would produce something called chromatograph, where you would see a color scheme where the dominant nucleotide at each position, which is now sorted by sides, left to right, can be called by seeing which of the nucleotide colors has the highest peak. So that was state of the art until as recently as 20 years ago. It could generate one sequence at a time, you know, fairly quickly, and it could generate something on the order of 1,000 nucleotides per run. So the question is this the process that would be used in forensics to identify a suspect? It depends on which forensics technique you want to uh, uh, talk about. Modern forensics is actually also moving to next generation sequencing, but yes, fundamentally there's some sort of sequencing and locus typing. Another question is, without initially knowing a viral sequence for an RNA strand, how do you determine the primer sequences? Good question, you don't. So what you do with next generation sequencing, you actually just share DNA and then you ligate known adapters to it, so it can sequence anything. I like this comment. Wish I had lived when this was used. I hate not understanding what's going on in the computers we use for all the next gen sequencing. I don't think very many people understand what's going on. It's very much a sort of a very niche domain. So people know individual bits and pieces, but there are very few that actually understand everything. Right, so there's a reaction mixture. And interestingly enough, this wasn't even the dominant sequencing approach at the time. Also in 1977, the Maxim and Gilbert published their sequencing technique, which now would be unthinkable. First, they used radioactive labeling. So this P32 is a radioactive label. So, you start with this. And the second thing you would do, you would also basically randomly digest it by cutting DNA molecules at random pieces using a specific nucleotide tag. Then again, generating, all the individual length of the sequence chopped at a random position. And you would tag it with this radioactive dye, which you could then run in a gel to separate things by length. And depending on, so the way to read this is that, you start from the end. So the last nucleotide is an A. So the full length of the sequence is 7. And you see, oh, this got cut by something that recognizes A or G, but it wasn't cut by something that recognizes G, so it has to be an A, and it's not a C or a T, so the first position is an A. Then you move down, oh, there's a length 6, so the last nucleotide there was cut by a C and a T, but not a C, so it's a T, and so on. So this is quite laborious, but this was actually the more popular technique until about mid-1980s, at which point the Sanger sequencing took over. So here's, I found this nice slide on SlideShare, which highlights the sort of major events in DNA sequencing. I mean, obviously, Mendel didn't know what DNA was. The canonical sort of discovery of DNA is, Crick, Watson and Crick, the double helical structure, which was reported and for which they received Nobel Prizes. And then as you go through, so the first the first sequencing in some sense started in the late 1960s. 1972, there was the first whole gene sequenced. So there's the Sanger a paper. Then 1984, a gentleman named Fritz Pohl discovered a way to not have to use radioactive tags for sequencing. So 1986 is basically the genesis of modern sequencing instrumentation, where applied biosystems, or ABI, developed first semi-automated DNA sequencing machines, and then it basically went faster and faster and faster. The first human chromosome sequence in 1999, model organisms, Arabidopsis and Drosophila, were published in 2000. The first human genome was published in 2001. And then in 2005, a fundamentally new technology was introduced. So this was the first massively parallel sequencer. And then it sort of took off. Then you have Illumina, which became and is the market dominant player at the moment that got into this area in 2007, and so on. So this is all very rapidly developing. And sort of the key points, the first modern sequencer was 1986. The next key point is 2005, where you had the first next generation sequencer. Now you have third and fourth generation sequencers. So this is progressing very quickly. There's another really nice slide courtesy to the National Human Genome Research Institute, which shows you how much it used to cost to sequence 1 megabase of DNA. So 1 million nucleotides. This is in dollars, right? So in 2001, it basically cost about $1 per nucleotide. So you were in the order of several $1 million to generate, one, say, chromosomal sequence. And then it sort of slowly goes down until 2005, 2006, when this is massively parallel sequencing is introduced. And all of a sudden, in a span of 5 years, you go from, $1 million. Sorry, that's not a million. That's 10,000, right? So I misread. So it's 10,000 here. I thought a million was too much, but it was 1,000 per megabase in 2007, and within 3 or 4 years, it was now 10 cents. And now you can sequence the entire human genome for well under $1,000 without data analysis and interpretation. So it's basically, the cost of sequencing has dropped by, 1, 2, 3, 4, 5, 6 orders of magnitude in the span of less than 20 years. So again, there's a really nice review that's linked here if you guys are interested in the history of sequencing. This was written by a bunch of folks that were very, very prominent in human genomics and other applications. So this was called, the 40th anniversary of sequencing, where we're going, where we are and where we're going. So first generation sequencing, Sanger, again, you start with genomic DNA, you chop it into smaller pieces, then you have to do sample preparation through cloning and amplification, then you do sequencing one sequence at a time. And it's indicated here that you basically terminate synthesis at each individual base and you read it out. This is one sequence per reaction. When you go to next generation sequencing, you've basically now used fluidics and high density chemistry and high density detection to run these types of reactions on a chip or a flow cell, millions of these reactions in parallel, right? So the actual technical details are very involved, but not terribly important. Just think about how this is, there's just miniaturized reactions. And sorry, and there's one other thing I would like to note here is that for these types of applications, long DNA is randomly shared or cut into shorter sequences because of technical limitations of the instrument. So for example, this strand, depending on how it's done, is usually only a couple of hundred nucleotides long, right? So if you want to sequence something very long, you cut it into pieces, you sequence the pieces, and you have to put them back together, which is actually quite complicated. With modern third or fourth generation sequencers, you don't actually do this anymore. You just take an entire long DNA molecule, pull it through this molecular detector. In this case, it's a nanopore, something that we've adapted from biological systems. Pores are in cellular membranes that allow things to go in and out. And basically it becomes a very, very sensitive current detection. As you pull DNA through, depending on which nucleotides are in the middle of this pore, and there's 5 or 6 of them, the charge will change and you get a time series readout. Obviously you do this, again in parallel, and this is all done using miniaturized technology, but you get a time series which you can then interpret and use computer software to predict which bases there are. So, and this will generate long DNA that can be read uninterrupted, right? So in principle, people have actually done that they generated sequences that are 1 million bases long, contiguous through these types of applications, which is quite remarkable. So staying with this overview, there are a couple of key concepts here. So when you're doing viral discovery or any other new discovery, you don't know what's there. So you sequence some large pool of DNA. Some of it is what you want. Some of it is contaminants, human DNA or environmental DNA. But you will start with a large number of short reads. Here they're colored again to indicate that each of these stripes is one nucleotide. Then you use very sophisticated and continuously developing computer software to patch them together. And the fundamental intuition here, I'm not going to get into it too much, is that you stitch things together based on the extent of sequence overlap. So if you sequence deeply enough, you can basically walk your way across the DNA molecule by patching pieces together that should be together, right? Again, that's hard to do in practical applications for a variety of reasons. Some of them are errors, some of them are, the fact that your reads are short, but there are standard solutions for dealing with this. Then you will assemble them into pieces that are as long as possible. They're called contigs. ideally, if your sequencing experiment was successful, the contig is the entire genome that you're trying to sequence. But a lot of times it will only be a partial genome. And then you have to use other techniques to connect these contigs together, paired end reads or optical mapping, again, not terribly important. And then you piece them all together into, say, if you're sequencing a large genome, into chromosomal arrangements, right? The nice thing about this is that you don't actually need to know what the outcome is. You can use just sequence overlap and some other clever tricks to lay out the entire or most of the DNA molecule without having any idea what it was. I mean, when people started assembling first sets of genomes, we didn't really know what to expect. I mean, we knew pieces, we knew individual genes, and maybe somewhat partial sequences, but not anything that resembled the final product. Once you have a reference genome, which we do for most of the interesting organisms, the problem becomes simpler. Now you're basically resequencing a known genome with some variation. So this is a cartoon that depicts variation in human genomes, right? So this is individuals 1 through 7.5 billion, give or take. And most of our DNA is exactly the same, but there are a couple of positions where there are individual differences between people. Or if you think of SARS-CoV-2 isolates, if you pull them from different infected people, by and large, those viruses will be identical, but they'll differ in a couple of key positions. So this is what you do here. You try to identify positions that are variable. All right, so that's shown here. You identify what's different and you count how many there are. And as you go across the genome, some of the positions will never change. Some of the positions will change rarely. Some of the positions will change frequently. And a lot of it is quite informative. We'll learn about it later in class. So, there are a number of different sequencing solutions on the market. And to understand on a very high level how to compare sequencers, you just basically need to think of this as any other kind of comparative shopping. It's a product. And if you want to do, if you want to make a decision about which one's right for you, you have to ask the right questions. And perhaps the first question that you should ask, what are the important features? Right, so how much data can it generate? All things being equal, perhaps the one that can generate the most data is the better one. What is the length of the read it can produce? For example, is it possible to use a particular sequencer to generate the entire SARS-CoV-2 genome? In principle. What are the error rates? I mean, you can generate a lot of data, but it can be garbage data, in which case it's useless. How quickly does it run? Right? Perhaps you need real-time diagnostics, so you need to be able to turn it around in a couple of hours. You can't afford to wait 2 or 3 days for the sequencer to finish. How easy is it to use? Do you need specialized equipment to run? Lab space, vibration-free environments, air conditioning specific environmental constraints. Do you need technical training to run these things, and so on? Then obviously, once this is decided, then the next obvious question is how much does it cost? And the other thing that you need to worry when you buy these lab instruments is how much does it cost to run? So an instrument itself might cost, say, $100,000, but then you need to pay another $250,000 every year to run it and service it. So you need to keep that in mind. And then finally, but perhaps most importantly, is how easy it is to analyze and interpret the results. Right. So if you have a great instrument that has really poor software that doesn't output anything you can use, then again, it's basically a brick. So here's just a very a fairly recent review on different types of sequencers that are available. The reference that I cite here is a much longer table, and the actual details are not that important. But for example, here are the things that are typically considered. So here's the manufacturer, Illumina. It's the biggest it's the biggest player in the market in terms of market share. They offer a whole range of instruments. You can think of them as car manufacturers in different models. So here's the compact and here's the luxury SUV. And as you will see, they will vary primarily in how much data they produce. So iSeq 100, which is the smallest and the cheapest one, will output about 4 million reads per run. Those reads are very short, so 36 base pair per read, which means that will output only about 150 million bases per run. To go to the very end of the extreme, this NovaSeq 6000, it generates 20 billion reads, which translates to about 3 terabases. So this is now 4 orders of magnitude different. And obviously the applications are also very different. So here it's small genome sequencing, targeted gene sequencing, and so on. And this NovaSeq is basically large scale human genome sequencing centers. The key observation for Illumina, so for one thing, they don't list prices because that wasn't a significant factor in this review, but the length of the individual reads, so for Illumina, they're quite short. So the shortest ones are 36 base pairs, so they're really quite short reads. And the longest ones go up to about 300 base pair. And Illumina also takes, fair bit of time to run. So the shortest turnaround is 9 hours. And if you go into these high-seq instruments, then it's basically now 3 days. Question is, what is it that makes a portion of viral genome more variable in comparison to other portions that remain constant? It is a very good question, but it's difficult to answer because a simple answer would be, selection and functional constraint, but I have to explain what that means. So some parts of the genome cannot afford to change because changing them will kill the virus. For example, if you're no longer able to bind to the ACE2 receptor, you're dead as a virus, so you can't do that. And some parts of the genome are quite variable, so if you start inserting mutations there, it doesn't change the protein, doesn't change a whole lot. But when you start delving into the details of these specific functional constraints, it becomes very complicated very fast. But we'll discuss that again. Probably next time we'll talk about intra-host variation and some of the selection that you see occurring on the SARS-CoV-2 genes. And here's the other instrument, and I chose these two fairly carefully because Illumina and Nanopore are the 2 instruments that are most frequently used for sequencing SARS-CoV-2, and they're sort of on the other ends of the spectrum. So Nanopore is a much smaller player. They sort of target a different end of the market. They all produce, a smaller number of reads, but these reads are very long, very long. So hundreds to thousands of kilobases, and they're actually quite cheap and easy to run. So a lot of the sequences that you see in public databases from SARS-CoV-2 have been generated using Nanopore. Which brings me to the next question that wasn't addressed in this table. What are the error rates? So instruments will always make errors when they sequence data. Simply, if you generate billions of bases, you're going to have millions of errors. But different instruments have very, very different error profiles. Question, epigenetic interrogation, does that refer to methylation, acetylation? Yeah, just regulatory gene modification, right? So histones, for example. But yeah, and it doesn't really apply to viruses as far as we know. There are some modifications, but they're not epigenetic in the human sense, so not methylated. But that's a good question. And then you also do it by sequencing. You actually do exactly the same sequencing protocol. All the differences are in how you prepare your sample. So you have to tag your DNA in a specific way and digest it and then interpret the reads in a different way. But the instrument that you use is exactly the same, and it just sequences DNA, but you process it differently. So there's a major difference in how accurate the sequencers are. So Illumina usually makes about 1 error in 1,000 bases, and Nanopore makes about 1 error in 7 bases. So obviously very, very different. And on its face, you might also look at this and say, how can I even use Nanopore data if it's so full of errors? I'll show you in a little bit how you can fix it in a very simple way. These instruments, they have software and computers that run them, either shipped by the manufacturer or plugged into an instrument. So the software that outputs bases will also generate a quality score, which has been standardized called Q-score or Phred score. Phred was a piece of software that was used to analyze Sanger chromatograms. So Q-scores are fairly old. They can be traced back to specific piece of software. But they're easy to interpret, and they provide a probabilistic estimate that the base produced by the sequencer is correct. So the actual formula for this is that you take the estimated probability of an error, take log base 10 of it, negate it, and get a Q-score. A very simple rule of thumb is that if you increase your Q-score by 10, you decrease the probability of error by a factor of 10. So Q-score of 10 means there's a probability that this particular base is incorrect that is 10%, so 1 in 10. You go from 10 to 20, the probability of an error is 1%. 20 to 30, it's now 0.1%. And 30 to 40, it's now 0.01%, and so on. Typically, Q-scores don't go past 50 because it's just, this point, very few instruments or applications either need this type of error, or this type of fidelity, or can generate it. So now that we have a little bit more of an understanding of what these quality scores are, let's go back to a screenshot of reads from the same data set that I opened with. And you can now see that for this particular output, the quality scores are quite high. So a score between 30 and 40 means that the probability that each of these bases is incorrect about 1 in 1,000. So if you take this at face value, the instrument tells you it's quite confident that the sequencing output that it produced is correct. If you look at a nanopore output, you will see a very different story. You will see that the quality scores are actually to the point of where the instrument doesn't know what's there. So quality score of 5 basically means almost 50-50. So there's very little certainty in the first or the second base, and then it goes up. And there are a couple of bases in the middle where the quality scores are in the high 20s, but it's a very, very different sort of degree of confidence of how of what the instrument thinks, or the software in the instrument infers the reliability of the data to be. So if you know nothing else, you should think to yourself that before you interpret these types of data, you have to be a lot more careful with how you deal with errors, and how confident or let's say how enthusiastic you want to be about interpreting the results that you derive from these data. Error correction, again, is a fairly involved area of bioinformatics, but the main intuition is very simple, and it's called error correction by wisdom of crowds, or majority rule. This is where the measure of coverage becomes very important. So if we assume that when the sequencer outputs DNA, the errors that it makes are independent, right? So they're not going to depend on where in the sequence you are or which bases you're sequencing. And by and large, this is not an unreasonable assumption. And if there are enough reads overlapping a given position in the genome, called coverage, then looking at each nucleotide and taking the most common nucleotide will correct most errors. Right, so here's an example. And if you want to, there's a little interactive document that I created. You can click on and play with on your own. But imagine here that you have a coverage of 1, which means this black sequence is the top. It's a sequence you're trying to determine. You generate 1 sequencing read, right? And your error rate for the sequencing read is set to be 5%. So about 1 position in 20 is incorrect. And for this particular realization of sequencing, there were 2 bases that were different, so T or a C. If this is the only read you have, there's no other information to tell you that these are incorrect. You might use some quality scores or whatnot, but you will not be able to tell without any other information that this T and this C are incorrect. Now, if you keep the same error rate, 5%, but all you do is now you sequence your underlying sequence multiple times, right? So you just, you shear your DNA and you have this part of the genome is covered by multiple individual DNA reads that were produced by the instrument independently, if you look at each individual read, none of them are error-free. They have 2 errors. Some of them actually have, 6 or 7 errors. But then what you need to do is that you need to look at each position vertically. So this C here, in particular, this C was always sequenced correctly. But if you go, say, to position 1, 2, 3, 4, 5, 6, there are 2 reads that were incorrect. So there were an A, but should have been a T. But there's enough of the reads that are correct to overcome these individual mistakes. And if you scan across the entire genome, you will see that none of the positions were incorrectly determined. If all you have to do is basically sequence the same thing 10 times, and this provides error correction because you assume that the errors are independently distributed. Now, if your errors for some reason all fell on the same position, for example, if sequencers have a really hard time determining when T follows T, then you would not be able to use this approach because then all the errors would bunch up and you have to do something else then. And in fact, this works reasonably well, even if you jack up the error rate to what the nanopore generates. So 15%. Now you look at individual reads, all these black positions are erroneous. But most of them are still corrected by the majority consensus rule, except for one where you have 3 Gs that now accidentally fell in the same position. But still, with a 15% error rate in individual reads and only 5 reads that cover the same position, you're basically down to only 1 error for recovering the true sequence that you generated. And you can even crank up the error rate to 50%. But if you have a coverage of 50, it still is enough to ensure that as a majority rule, you recover the correct sequence. Now, I don't have the slide. I'm not going to go back to it, but the SRA metadata that I showed on the second and the third slide showed there was a coverage of 6,000, right? An average coverage, which basically means that each position is covered by 6,000 independent reads, which should indicate to you that if this is not uniform and some of them are still going to be, there's still going to be some errors, but by and large, with this much sequencing depth, just using this idea of majority rule, you'll be able to recover the true sequence in the vast majority of genomic positions. All right, so as a rule of thumb, when you're doing human genome sequencing, coverage of 30 is considered to be very, very good. You don't really need to do more than 30 times over the entire genomic sequence to correct most of the errors. In fact, it's an overkill in a lot of applications. All right, so now we've generated a sequence. We've done something to correct errors, perhaps. And we now have a large file with many DNA sequences. So the next question is, how do you find out what you sequence? Well, there are a couple of steps there. What causes this difference or variability in the error rate? There are particular motifs in the DNA sequence that, depending on the instrument, that will have an easier or harder time with. It really depends on the platform. So for example, Illumina sequencer will generally have higher error rates towards the end of the reads because of the chemistry that they have to go through to generate the sequencers. Some of the earlier instruments like 454, which was the first next generation sequencing next generation sequencing platform, had a really hard time detecting genomes or calling genomes that had a bunch of the same nucleotide in a row. So if you had like 5 or 6 A's in a row, it would know that there were several A's, but it would have a very difficult time distinguishing 4 from 5 or 6, right? So that would be a sort of systematic error. But it depends a lot on the underlying platform, and it's pretty well characterized. So we have a good idea of which types of errors are random, which ones are not, and you can try to correct for them in a variety of ways. So you take this very large file produced by a sequencer, then you assemble your reads into longer contigs using overlap or something along those lines, And these contigs you can think of as genomes or partial genomes. So now you have longer sequences. Once you've done this, again, on their own, they're not terribly informative. But what makes them very informative now is that you can query them against databases of known organisms to identify what they look like. So once you've done this, you can also go on and annotate genomes by features. So try to identify genes or specific motifs like these polybasic cleavage sites, things that we know by studying other sequences. And always look up similar features in databases. So the power of bioinformatics and comparative genomics is that we have such a vast reference database that almost nothing that we sequence is going to be unidentifiable. There's going to be something similar to it somewhere. And if you're doing resequencing, you know what you put in, right? So if you were sequencing SARS-CoV-2, or if you're sequencing human genomes, you should get that back. There's a great a resource maintained by the Howard Hughes Medical Institute that's linked here, that if you're interested in a very introductory sort of module of how DNA genomes, or how DNA is assembled to genomes, I highly recommend this. But in pictures, what you have is you have DNA clones. This is what you're trying to sequence. You have to randomly cut them into short pieces, sequence them, and then there's this magic black box bioinformatics that converts the reads back into the contiguous genome that they came from. And all you're really doing here, again, is using some idea that's based on overlap between reads to stitch them together. You create islands of things that you can stitch together into contigs. If you're lucky enough, you'll be done. If you're not lucky, if you're missing some of the reads for whatever reason, then you have to do something else, an additional layer of sequencing to order these contigs together. And then if there's still gaps or things don't fit together, they need to do additional sequencing to fill in the gaps. And then you produce a draft genome, which is annotated and may have remaining gaps which will later be filled in. In fact, if you look at the SARS-CoV-2 genome, it has undergone several revisions. They're fairly minor. If you look at the human genome, I think it's now in revision 38 or 39. So when the first human genome that was published was missing a significant part of the genome, and there were a large number of fairly significant errors in it, which since have been corrected and will continue to be corrected. And one of the things you can also help yourself with is you can make your analysis much simpler by doing more work at sample prep. So if you've done any kind of photography, I tend to think of this as digital zoom versus optical zoom, right? So you can, and you can try it on your, even phones have it. So digital zoom basically uses software to create an image that is a higher magnification that what your sensors allow you to do. And optical zoom uses, better optics, heavier lenses or more lenses to zoom in the image. And typically, if you do the optical zoom, if you use physics to enhance the image, it'll be better than if you use software to do this. So a lot of the same ideas go into sample prep. If you make things cleaner before you sequence them, it's much easier to deal with the analysis. Question, are epigenetic changes accounted for in any of the public genomic sequences, or does it vary too much? Oh yeah, there's plenty of epigenetic annotation. So there are separate databases for this, and typically will not be a part of the genomic record. It's considered to be a higher level variation. It's actually also going to vary from individual to individual. So it's there, but it doesn't sort of belong necessarily in the reference genome. We'll talk about human genomics in a couple of lectures, so I'll mention it then as well, because it's not, we don't want to be just about the virus. We want to learn about the host as well. In this particular study, and we're still sort of unraveling this RNA metatranscriptomics, so viruses are sampled together with host and other DNA, right? Because you're pulling out a biomedical sample from somebody's lungs or blood or saliva, whatever it is that you're trying to sample it. And the assumption is if you took it from a human, most of the DNA that's there is going to be human. So it's going to be much more abundant than viral DNA. And here you can do the digital or the optical analog. So the digital analog would be you sequence everything, including the human DNA, and then subtract the host DNA computationally. Or you could do sample prep and amplify or filter non-host DNA before sequencing. So you basically don't put host sequencing on the DNA. Host DNA on the sequencer. And you can see for this particular data set, going back to the original discovery paper where the reference genome came from, is that their SRA record is public, so you can go and look at it to see what they actually generated. So in this case, they have 8 gigabases. This was done using metatranscriptomics, using an Illumina platform, using RNA sequencing, so random selection, so random DNA amplification. This is paired end reads. It shows you that it came from human lung metagenome, and then you can click on another tab, which is if you look at the experiment, you can actually have GenBank display what organisms are represented by those reads. So go back for a second. There is 28.3 million reads, of which 61% are annotated as dark matter, so they don't map to anything. So basically, 60% of the read of this data set is has no relevance to viral discovery. And in fact, you start drilling down. There's bacteria, cellular organisms, bacteriophages. There's this tiny little sliver of 0.2% of all the reads that map to viruses. And some of those map to things other than SARS-CoV-2. So it's worth stopping here and thinking for a second that the way this discovery was done, it really is the brute force approach. You sequenced millions upon millions of reads, and then you ended up throwing about 999 out of each 1,000 because they were not useful, they were not viral reads. But there was enough left over that you could actually not only identify there was a new virus, but produce an entire genome that wasn't actually too bad. This isn't quite the entire story because there's a standard protocol for doing this. And I'll show you just a few key points. It is very, very important for any sequencing experiment to prepare your sample properly. So the first thing you do is you want to sample the right biological compartment. So in this particular case, you had somebody with pneumonia that you suspected was viral. So the logical place to sample would be from the lungs. There's a standard sampling technique called BALF, bronchoalveolar lavage. It's a diagnostic method of the lower respiratory system in which a bronchoscope is passed through the mouth or nose into an appropriate airway in the lungs with a measured amount of fluid introduced and then collected for examination. So you go to the source of the infection. You don't try to draw blood or saliva because then you would have even less virus. Then use a standard RNA sequencing kit. So that's produced by large biotech companies. I don't remember who it was, it might have been QIAGEN, but it's shown in the paper, you can see what it is. So this is standard, it's basically a kit, you just follow protocol, right? If you're a trained lab scientist, you can do this. Then you do some purification, so you remove RNA that is ribosomal, so it could not possibly be viral. So you do basically a sample level filtering, so you don't sequence RNA that is useless. Then once you filtered this ribosomal RNA, you do paired end, 150 base pair reads using the mini-seq platform. You can go back to the table and look up what mini-seq platform is like. It's sort of, it's not high end. It's kind of towards the lower end of the spectrum, so you don't need that much data for viruses. Once was all said and done, there were 56.5 million reads that were just assembled de novo, right? So they just put it into a piece of software and said, find what's there. They used two pieces of software, one called MegaHit, the other's called Trinity. So MegaHit generated 385,000 possible assembled contigs that ranged in length from 200 to 30,500 nucleotides. Then Trinity generated a lot more contigs, but they were shorter. So depends on which software you use and which settings you use. So now you've basically reduced 56 million reads to either 385,000 reads that are all stitched together, or 1.3 million reads. So this is a computational filtering step. At this point, you take this and filter it against the human genome. So basically see, do any of these sequences look like they came from a human? And if they did, you throw them out. So this is computational subtraction. And you do this by querying a database of the entire human genome using a specialized piece of software to do it quickly. Bow tie short read mapping. And then you compare the longest contigs that remain to the database. And in this case, what they did, they actually took the longest contigs, so this 30.5 kilobases and 11.7 kilobases. And when you ran it through a database, it came back looking like SARS, but not SARS. So at this point, they said, we have something that makes sense. It's an RNA virus. It looks like something that causes viral pneumonia, and it is new. So that could be our thing. And then you have to do confirmation, right? So that's not enough. Then you actually go on and say, now that I have a candidate genome, I'm going to design primers for direct amplification, so PCR amplification of this genome. I need to be able to confirm that it's there by doing independent amplification and characterization of this genome from the same sample. And then once you've done this, so now you actually, instead of looking for the proverbial needle in a haystack, where you're looking at 0.2% of all the viral reads in a very large collection of genomic data, now you specifically amplify and sequence only the viral genome. And then once you've done this, you can refine the genome and you can perform detailed comparative analysis with respect to similar coronaviruses. So this is basically how viral discovery is done at a very high level. All right, so you do high throughput sequencing. If it's an RNA virus, you do transcriptomics. So you have to do RNA sequencing. Then you do a lot of heavy duty computational lifting to assemble these short reads into longer reads, filter them, throw away human DNA and everything else that is not a virus. Then you come up with a candidate genome, and then you have to do additional confirmation in order to establish for sure that you have that you found something that's real. Right? Because you could also have a computational chimera. You could have a software product that doesn't exist in nature just because the reads were not assembled properly. All right, so, but this is a very, very important first step. Once you have a reference genome, it enables all kinds of other things like resequencing and molecular diagnostics, which is where we're going to go next. Here, let me pause here for a second, take a few sips of coffee and see if you guys have any questions. All right. So, again, every biology major should know what PCR is, but I found this very pretty slide just to make sure that we have all the correct terminology for what we need for the rest of the lecture. And again, this is courtesy of the National Human Genome Research Institute. So PCR, again, is a foundational technique that allows you to amplify, meaning make lots and lots of identical or nearly identical copies of whatever it is that you're trying to amplify. So you start with a very small number of input DNA molecules, and then you have to selectively copy them. And the way you do this is that you use natural process of polymerization and template extension, which happens in nature all the time. But you have to guide it to amplify only the sequence that you want to amplify. And you do this by developing specific primers, which we'll discuss in a second, because they're very, primer design is very important for diagnostic and performance, and it's sort of bread and butter of sequencing and molecular diagnostics. And you always have to, so this, you always have to keep in mind that we're doing double-stranded processes. So you need, for double-stranded DNA, you always need to have forward and reverse primers. So, yellow and magenta here. So 5' is left, 3' is right, forward strain, reverse strain. And you start with, and it's important. So, there's a nice visual clue here where the blue strand is what you're trying to amplify. So this is the input DNA. So you tag it with a primer. Because there's a primer, now you have a polymerase that's going to attach to this primer and start synthesizing a complementary strand right to left on this strand, and then left to right on this strand. So now you've made 2 copies. So what you do next is that you denature them. So you heat up this, the bath in which this is all happening. So what were 2 strands become 4 individual templates, and then you cool it down to an appropriate temperature where this polymerization can occur. Now you go up to 4 copies, 8 copies, and then you go 20 to 30 cycles. And every time you double the number of copies, and at the end you start at what was 1 template is now 2 to the 20 is 1,000,000, 2 to the 30 is close to 1 billion copies. So this is biological amplification. And in order to amplify, and there are obviously a lot of trickery involved here. So PCR, even though it's a very established technique, there are a lot of variants of it, and it can very easily be done poorly. And if it's done poorly, you can have all kinds of things that you don't want to happen. You can have polymerization errors, you can have things that recombine, you can have poor amplification, and so on. But when somebody tells you today that they did a nucleic acid test for SARS-CoV-2 or anything that is a virus, it is based on the PCR test. So there are primers involved, and there's this step involved when you take a small amount of viral DNA and you amplify it. So when you design a primer, because this is something that molecular biologists have been doing for 40 years, there are manuals and cookbooks for how to do this properly. There's software tools that allow you to avoid mistakes and select primers that are appropriate for your template. But you always need a pair of primers. You need the 5' and the 3' primer, forward and reverse. They must be designed to follow a large but well understood number of guidelines. So the one is that they must be specific. So they have to be a part of the sequence that you're trying to amplify. So they should bind only to the sequence that you want to amplify. And they should also be sensitive, so bind to enough sequences that you want to amplify. If they're non-specific, if they bind to everything that's in the solution, what's going to come out at the end is just a lot of random DNA. Some of it will be what you want, but a lot of it will be what you don't want. If they're not sensitive, you'll have no product. Sorry, specificity, I say sensitivity, but specificity can be improved by nested PCR and other techniques. So if you want to be very sure that you're amplifying what you want, you can actually have multiple variations of the technique. It should possess other properties that improve reaction. And there are a number of computational tools, for example, Primer Blast that's maintained by NCBI that allows you to input a sequence and out will come some sets of primers. If you're interested, there's a Cold Spring Harbor Lab Protocols paper that I linked here that describes in a lot of detail which primer features that are desirable. So for example, a primer should be of a specific length range. So 18 to 30 bases is optimal for most PCR applications. If you make them shorter, they'll be non-specific, so they'll amplify everything. Longer primers, because it's an RNA molecule, it's a nucleic acid molecule. It can have multiple, it can have secondary structures. So you have, it can fold on itself, can create stems and loops and slow the reaction. There's a very specific temperature range in which this to happen. So your primers have to melt between 55 and 60 degrees Celsius. And the forward and the reverse primer also need to melt at about the same temperature. Amplicon melting temperature. So once you've made your DNA copy, then you have to denature it to go into the next cycle. So they must melt at 92 degrees. Celsius, which limits the amplicon length to about 600 base pair. So it's actually difficult to amplify something that's outside that range. And secondary structures on primers should also avoid secondary structures like 5' and 3' complementarity, so they can bind to each other and clog up the reaction. So here's an example of, so I just out of curiosity, I took the spike gene of SARS-CoV-2 from the reference genome and fed it into the NCBI tool and said, just find me some sets of overlapping primers. And here's what it produced. It said, here's a collection of, say, 10 primers that put together cover most of the genome. Right? That would be one possibility to define how you can amplify the viral genome. But other things are really obvious here. If you want to amplify something that's long, so the spike protein is coded for by about 3,600 nucleotides or 3,819 nucleotides, you cannot amplify it as one amplicon. You have to chop it into individual pieces. So once you have these, once you have a reference genome, you can use computational and other comparative techniques to design a set of primers that will amplify pieces of the genome, right? And for this, you must know the genome. So you need to know what you amplify. And ideally, you need to know more than one genome because you want to have the same set of primers that will amplify most viruses, even if they change in other positions. So once the viral genome is known, sequencing of new viral isolates can proceed by amplifying fragments of the genome and mapping to the existing genome to identify differences. So this is picture C here from this PLOS1 reference, where you start with your genomic template. You do PCR from complementary DNA, so it'll come back as individual amplicon pieces. So they don't, they're all shorter than the genome, but they will presumably overlap and represent the entire genome. This creates amplicon libraries, which you can then put into a sequencer and generate. And here's an example of why you need to have overlapping primers, because if you don't have overlapping primers, there are parts of the genome that are not going to be sequenced. Like here you have this sequencing coverage, and the two dotted lines are where your primers are. And if you don't have overlapping primers, you won't be able to sequence, say these areas where there's a dip in coverage. Now, fortunately for us, prior to the emergence of SARS-CoV-2, there was a group set up. They had actually started work, I want to say in 2013, 2014, during the last outbreak of Ebola. They're called the Arctic Network. They're primarily based in the UK, but they're participants from a number of academic and government institutions worldwide, in the US as well. And their goal was to develop an end-to-end system for processing samples from viral outbreaks to generate real-time epidemiological information. Their specific target was fast-evolving RNA viruses. So things like Ebola, SARS-CoV-2, influenza. The important features of these viruses that need to be acknowledged is that they continually accumulate changes in their genome. And that both creates a challenge in the sense that you need to have a sequencing protocol that is able to tolerate these changes and still amplify the genome and sequence it. But these changes are quite useful to reconstruct the immunological processes that drive the epidemic. In fact, like that Nature paper that you guys read for after the first lecture, used comparative analysis. And last time we discussed comparative analysis, which requires you to have multiple genomes that are different from each other. So you need to generate more than one genome to learn something about the virus. You need to generate many, many, many genomes. And their approach was to use the Oxford Nanopore technology, specifically the MinION instrument, to develop what they called a lab in a suitcase. And their sort of aspirational goal is to be able to go to wherever the outbreak is. SARS-CoV-2 obviously is everywhere now, so you don't have to go to the outbreak. It came to us. But if you have an outbreak in a remote location, say in sub-Saharan Africa or in Southeast Asia or somewhere else, you want to be able to take the instrument to that location and not necessarily wait for the sample to come to a centralized lab somewhere in Europe or the United States or in Asia. So they emphasize portability. They also designed and validated very early on in January a standardized set of primers for SARS-CoV-2 amplification. It is publicly available. You can go and look them up. They're available in GitHub. In addition to the primers, they also provide a full laboratory protocol. And because it's standardized, tested, and sort of community sourced and validated, it has become very popular for SARS-CoV-2 sequencing. It is not the only one. You have reference labs and large institutions that are able and have the technical expertise to do their own sequencing, design their own primers, and use their own lab protocols. But for many smaller contributors, especially the ones that publish sequences in this database, GISAID, from all over the world, they use Arctic primers and Arctic protocols. And I think this is a great example of what you could call laboratory open science. So both protocols and primers are available online. So there is a website that contains laboratory protocols called Protocols.io. You can go and find their version 3 protocol. It's going to give you a very, very specific set of instructions about how you do DNA or RNA prep and sequencing. Complementary to this, there's a GitHub page. GitHub, if you're not familiar, is the preeminent location for software development and source code sharing, but it's just an easy to use resource when you can have version documents, and they're available for everyone. So for instance, there you can look at their primers, and they're right here. You can see that left and right is forward and reverse, and here are the actual primers, the length, and TM is the melting temperature. So you will see that they are in the sweet spot that I mentioned a couple of slides ago, between 60 and 63 degrees Celsius. So the other great thing about SARS-CoV-2 is that a lot of a community effort went into testing and improving these protocols. So for example, the Arctic protocols on the previous page were version 3, which implies obviously that there was a version 1 and a version 2. And the reason there's a version 3 is because there was a paper, well, it's a preprint. It might have been published, but I couldn't find the published version of it. The preprint is referenced here. What they've observed in this experiment is that, this is a coverage plot. So let's walk through it. So what they've done here is they've demonstrated, 2 experiments. On the x-axis, you have the coordinates of the SARS-CoV-2 genome from 5' position 0 to 3' position 30. These little boxes are overlapping amplicons. So you can see that the Arctic primer set, I think there's close to 100 primers. So you generate about 100 primer sets. You generate about 100 amplicons that taken together cover the entire viral genome, and then you map them onto the reference genome and call variants. And what's shown here is the coverage. So what they've done is they've done a whole sequencing protocol. They took a positive control sample with SARS-CoV-2. They've run through the experimental protocol. They generated 100 amplicons. They sequenced them all, and they mapped them back into the genome to see which ones were successfully amplified or not. And the coverage, just to remind you, means that after they've done the sequencing, they were able to say that, for example, for this region, on average, between 100 and 1,000 reads covered this part of the genome. So all of these primer sets successfully amplified their amplicons. But then if you go, say, into primer 18, or just generally the ones that are shown in red, you have these dips. So you have much fewer reads mapping to these amplicons, which could mean that the reads weren't there, and that's the assumption that they're making, that these primers were not very good at amplifying their specific primers. So there's their specific amplicons, so the 7, 9, 15, 18, 21, and so on. And then there's some diagnostics here about how this was done. So how many PCR cycles were done, and how much sequencing output there was. But they basically did this study and went back to the Arctic people and said, look, some of your primers aren't working too well, so let's look into this and revise them. And then following additional analysis, they identified some of the issues. For example, some of the primers are cell hybridizing, which is something you should be avoiding. So the primer set was revised and improved. So here's a screenshot from a white paper from the Arctic Protocol website, which basically does a different take on the same type of data that I showed you in the previous slide, where you have, so on the axes are swapped here, and there's no genome, so you have individual amplicons, right? So presume they are from the 5' end of the genome to the 3' end. So each one of those is a couple of hundred nucleotides that's being amplified. And what you see on the x-axis is the coverage depth, right? So how many reads were successfully generated from a particular amplicon, and 50 is what they consider to be sufficient coverage. And you can see that basically what you want to avoid is you want to avoid situations where a particular primer fails to amplify enough reads to reach this minimum coverage. And you can see that in version 1, there's this primer that looks like number 76 had no reads whatsoever. There's another primer that's around 20 that had no reads whatsoever. And there were 2 primers, one here, sorry, primer sets, and one here that failed to reach their coverage. Right, so in version 2, they successfully fixed this primer, but introduced, and this primer, I said, but they introduced another error, and there's still some dips. And in version 3, they were finally able to get everything sufficiently even to the point where there are no issues in coverage. And version 3 is the one that's currently being used in the Arctic network. All right, so again, and none of this is unique to SARS-CoV-2. What is unique to SARS-CoV-2, I think, is the open source, open collaboration flavor or aspect of it, especially the contribution of folks in the Arctic network that were forward-looking, and we're kind of ready to go very early. So in large part due to their efforts, we had so many sequences deposited, generated and deposited so quickly. So that's unprecedented as far as outbreak surveillance goes. And it was important to develop a validated and standardized set of primers and laboratory protocols for this to work. All right, so moving on to the next and final topic of today's lecture, which I think kind of nicely segues from PCR amplification and ampliconic sequencing. This would be SARS-CoV-2 diagnostic testing. So here's a recent review in JAMA that I cite that is sort of very accessible if you're interested in getting a little bit of a background on this. We'll go into testing, a lot more, probably 2 lectures from now, specifically, different types of tests. But there are a couple of key insights that you should learn about testing and what it can and cannot do. So first, there are 2 fundamentally different types of testing. So there's nucleic acid testing, which is based in PCR. And that is usually more sensitive, and it's able to detect actively replicating virus, which means it is specifically designed to capture early or active infection. There's a whole other set of testing for antibody or antigen testing that don't look for virus, but actually look for traces left by the virus, either the antigens or antibodies that were raised in response to the virus. They're very, very different tests, completely, different chemistry and different logic, and they test for different things. So if you have an antibody to a virus, it could mean that you still have some of the virus, but it could also mean that the virus is long gone and you just have the antibody that is left behind. So if you look at the picture that I took from the JAMA review, is that you, as time 0, you have the onset of symptoms, which is when the patient realizes that they're sick. Prior to this, there's an incubation period. There's going to be, as you know, quarantine for 14 days. So, which is why you have, 2 weeks exposure. So you had exposure sometime in week -2. Then the virus was, replicating, but not apparent to the patient. So you didn't know that it was. If the patient was tested prior to the infection, what happens here is that the viral load and the number of copies of the virus increases with time. So very early on, there's so little virus that even the most sensitive test won't be able to detect it. As the viral titers increase, you'll be able to see some of the tests coming up and being able to detect it. Once the symptoms hit, you sort of have the peak level of the virus. So this is the quote unquote sweet spot for a nucleic acid test because there's so much viral RNA. Then as your immune system starts clearing the virus, these tests become less able to detect infection because there's less virus. But the other ones that are based on antibody are following the opposite trajectory in that they're more able to detect the presence of an antibody. And you can also have different types of PCR sampled from, either nasopharyngeal swab PCR, virus isolation from respiratory tract, so BALF, and stool PCR, and then you have 2 different type, IgM and IgG antibody, which we'll discuss later. So you might ask how many different tests there are. And as of yesterday, there are 160. In fact, one of the tests was approved 2 days ago on the 8th of September. So there is an FDA website, which is linked here. You can look at it. And there's a searchable interactive list of tests that have been approved, emergency approved, most of them. And here's a partial table of these. I sorted them by the date of approval. You can see a summary for each of the tests. So the manufacturer, some of them you might recognize like Roche, Molecular Systems. A lot of them I've never heard of before because there are a lot of people getting into the area of testing. There's obviously, a lot of tests that could be done. And this is both important and commercially attractive area of development. The name of the test is shown here. So, for example, you can see some of them, like the Roche test is a combined test, so a test for SARS-CoV-2 and influenza A and B. The technology is molecular, so I've filtered it on molecular, so it's all based in PCR. Authorized settings, so age basically means healthcare settings, so it has to be done in a lab. Authorization documents is healthcare providers, patients, and so on. So, question is, how long do the antibodies for COVID last after exposure? Yes, there's a lot of speculation in the media about how long they last. It's actually not terribly clear. There's no reason to believe, and we'll discuss it again later when we talk about the immune response to SARS-CoV-2 and other diseases. There's no particular reason for the antibodies to not be, to decay quickly. But I think there's a lot of, because there's so much interest in it and there's so much writing on, say, protective immunity, that people read too much in what's out there. So most of the scientists that do these studies, you will see they're trying to be very cautious in interpreting their findings. So we don't really know at the moment. And there are different types of immunity. There's T-cell immunity, B-cell immunity, and innate immunity. They're all important. They're all different. So we'll talk about them. But it's hard to say. We'll measure it. We've been measuring it for influenza, for instance. And for influenza, it can last easily years, if the strain doesn't change too much. But the problem with influenza is that the virus changes. And, what used to work 5 years ago doesn't. It's quite complicated. There's a lot about the immune system that we don't understand, unfortunately. So maybe SARS-CoV-2 will push us forward in that area. You can look, now that I encourage you to read government documents, but if you want to be an informed consumer, this is something that you might want to look at. If somebody says, I'm going to, I want to get a test for SARS-CoV-2, you should be knowledgeable enough to go, say, to the FDA website and try to see what this test was actually approved to do and what it is that it cannot tell you. So a lot of it will actually be quite specific. So here's an example of one type of approval. So your product is a qualitative test for the detection. So qualitative is an important word. It just means yes or no. It's not really designed to test how much SARS-CoV-2 there is in different types of swabs. The SARS-CoV-2 nucleic acid is generally detectable in respiratory specimens during the acute phase of infection. Positive results are indicative of the presence of SARS-CoV-2 nucleic acid. Clinical correlation with patient history and other diagnostic information is necessary to determine patient infection status. Positive results do not rule out bacterial infection or co-infection with other viruses. Negative results do not preclude SARS-CoV-2 infection and should not be used as the sole basis for patient management decisions. Negative results must be combined with clinical observations, patient history, and epidemiological information. Then there's a specific set of requirements for this test to be used. So to use your product, SARS-CoV-2 nucleic acid is first extracted from specimens. The extracted nucleic acid is then reverse transcribed into cDNA, following PCR amplification detection using an authorized real-time PCR instrument. There's specific types of lab supplies and instruments you're able to use. And then a lot of these tests, in order for them to work properly, you need to have positive controls, negative controls, internal extraction controls. And this is all very important because it significantly reduces the rate of errors of individual tests. And it specifies for each individual protocol, which tests are provided and which ones are required. Right. So you can look at these documents if you're interested. Surprisingly, it's actually very difficult. If you get a test, you probably won't know which specific test you're being offered. But I think it is important to know because they're all very, very different. And the vast majority of the tests you're likely to encounter as a consumer or a healthcare patient would be based in quantitative reverse transcription PCR. Which is actually a very general and very powerful technique to both detect and quantify the amount of input DNA. So the RT step is necessary to convert viral RNA into DNA. So this is reverse transcription. And most of the tests that you will see are one step. So they do RT-PCR and amplification in one step. So you don't have to separate them. The two-step would do RT-PCR first. We'll do RT first and then PCR second. It's a highly sensitive set of techniques to amplify and quantify a specific nucleic acid sequence with detection of the PCR product in real time. Quantification, it basically can also tell you not if there is some virus, but how much there is, which is important, as you will learn from the reading assignment today. Quantification is achieved by determining the cycle when the PCR product can be first detected. And this is in contrast with sort of traditional endpoint detection conventional PCR, which does not enable accurate quantification of nucleic acids. So this is all based on actually a great resource from QIAGEN, which is a commercial producer of a lot of the reagents and kits for RT-PCR. You might have, a couple of months ago, QIAGEN was actually running out of supplies because so many people were using their kits for testing. So one of the issues that I'm not going to discuss in detail, just mention, is that what SARS-CoV-2 revealed is a lot of the really tedious and mundane aspects of healthcare and diagnostics. So supply chains, just having enough reagents on hand, being able to scale up production of things like PCR reagents, and a lot of the systems were stressed, and some of them did better than others. But QIAGEN, for example, was stressed by demand on their kits because it really went through the roof. So the interesting thing about RT-PCR, about quantitative PCR, is that it has one more, flavors of it have another part of DNA that is important, so-called probe. So quantitative PCR is based on light detection. And again, you want to detect and quantify the amount of input DNA. So you start with PCR primers, just like for normal PCR. So forward and reverse. And the sequence that you are amplifying can also contain a probe, so a specific sequence between the primers for fluorescent detection. And detection is performed using an optical readout once enough PCR product is accumulated. So there are two major ways to label PCR product. One is called cyber green. And cyber green is a compound, it's a fluorophore that will bind any double-stranded DNA. So here's a picture from this. So you start with your template, 3' to 5'. There's some cyber green in the solution. It's not doing anything. And then the primer hybridizes, and then you start elongation. So now you have double-stranded DNA. Cyber green, if it's bound to double-stranded DNA, and if you shine the right wavelength of light at it, it'll emit a detectable fluorophore, which you can then read off with a different detector, so blue in, red out. And as you synthesize more and more and more molecules, so double-stranded DNA is the presence of a successful hybridization. You don't know what double-stranded DNA it is, so hopefully it's the one that you are trying to amplify, but in principle it could be something else because it doesn't recognize any particular motifs, just that it's double-stranded DNA. But if you make more, with each cycle you have more and more DNA, more and more readout, and you'll be able to detect sort of stronger and stronger signal as you continue through the reaction. This is actually not used that much for SARS-CoV-2 detection. There has been some literature on how to use it, and the primary concern with it is it's somewhat non-specific. So the second one is to use TACMAN probes, which means that you now have a third nucleic acid motif. So now you have, recall, this is the template you're trying to amplify and detect. So you have two primers. You also now have a specific landing pad or a probe between these two primers in the sequence that you're trying to characterize. And you have these probes, complementary probes, that can bind in the middle of your sequence that have a fluorophore and a quencher. So they're basically fluorescently labeled molecules. So they bind only to what you're trying to amplify. And if it's a single-stranded, if it hasn't been extended by PCR, it's designed in such a way that you have two molecules. You have the fluorophore, but you also have a quencher. So unless something else happens, shining a laser light into this fluorophore is not going to detect, is not going to cause any emissions because whatever it emits will be absorbed by the quencher. Now what happens here is that once the primer gets to where the probe is, it cleaves off the probe. The probe is now free to float away from the quencher. And when you have the right wavelength of light coming in, you'll have detectable emission. And again, the reason for this happening is you actually want to see active elongation. So not only did you have binding, but you also had a successful primer attachment, and this was being actively extended by the polymerases. So it's important to realize that the TACMAN probe principle also has a probe or specific sequence for fluorescent detection, which is performed using, how does the primer cleave the probe? I don't remember the actual details, but if you look at the, the QIAGEN document, it gives you specific details. It also depends on the design of the probe, but it's a well-established chemical principle. I'm not really a biochemist, so I know just enough not to get myself in trouble. The other nice thing about TACMAN PCR is that probes can be multiplexed. So you can have multiple populations that you're trying to amplify when, if you have fluorophores that are emitting different wavelength, different wavelength of light, so red, green, blue, you can detect multiple components. And one of the important things that you will see if you read any literature about RT-PCR detection, there'll be this number called CT, or characteristic threshold, which is basically how many PCR cycles have to occur before you're able to detect signal, right? So remember, and you'll see these very characteristic curves. If your PCR experiment is designed properly, you start with a very small amount of DNA here, cycle time 0. There's a threshold that is a limited detection. So your optics are not able to see any light until there's enough fluorophores being released by hybridization and elongation that they make a strong enough signal. And even though you're doubling the number of DNA molecules every time, initially there's not enough signal for this to be detected. And then you reach a critical threshold, which is called the CT, at which point you're able to see the light. And then for the next few cycles, you basically have this nice doubling, right? So the light intensity becomes basically twice as bright with each PCR cycle. And then you run into the plateau because you basically run out of reactions. Run out of reagents, or you saturate the reaction in some other way. And you can do very simple, basically, slope estimation with known concentration measurements to extrapolate or infer how much DNA was in your sample based on what cycle number and how quickly it was being amplified, which is where the quantitative part comes from. The real time is basically you're watching it with every cycle, and at some point you're starting to see the light. And then the quantitative means that you can use the rate of growth in this linear phase to estimate how much RNA was there to begin with. All right, so this is true of any TaqMan-based qPCR assay, and there are a number of them on the market. Like I mentioned, there were 160 as of yesterday in the FDA database. But let's go back a little bit, further than that to February of 2020, where there was a nice review paper that compared at the time how did different diagnostic primers and probes performed in identifying the presence of SARS-CoV-2 nucleic acid. So this was done in February 2020. And there's a really great depiction of where all these primer and probe pairs were located. So you will see that they are identified. So there are 3 ingredients to each diagnostic test. So you have 2 primers. You have the forward primer, the reverse primer, and in between them you have this square, which is the probe. It shows you which parts of the genome in reference genome coordinates are being targeted. So they're targeting different genes. You notice that there is a large number of probes in the nucleoprotein gene. You might ask yourself, how would you design a probe and primer set? And one of the considerations that you want to put in this design is you want whatever it is that you're targeting to not be able to change very quickly. So you will usually look at structural proteins that are known to be fairly conserved from other related viruses. So for instance, one of the things you could do is say, this is a SARS-like virus. So we had designed primers back in 2003, 2004 to study the original SARS, and it was a nucleoprotein, and we have nucleoprotein probes in other coronaviruses, so let's put something there. And that's pretty much the only information you have to go on, because you don't have a whole lot of data on SARS-CoV-2. So this is February of 2020. There are not that many sequences. So you can't wait and see how the virus is evolving to decide whether or not this is a conserved or variable region. So you have to make a good educated guess. So for instance, one of the reasons this probe is here is if you remember the slippage site, which is unique to SARS coronaviruses, that's the area of the slippage site, which has very, very strongly conserved secondary RNA structure and other constraints. But potentially because of that, it could also be a poor probe because of the old secondary structure. Right? So with that out of the way, let's see what they've actually evaluated. So their goal was to do a standardized experiment in which they compared how well each of these primers performed at detecting SARS-CoV-2 infection. So you have nucleoprotein probes from China, Hong Kong, Japan, Thailand, 3 sets of probes from the CDC, so from the US. And again, for each of these, you have 3 components. You have forward primer, reverse primer, and probe. You can see these are all short nucleotide sequences. If you remember, the constraints are given to you by the PCR reaction and just general RNA chemistry. There are positions in the genome where these are located, and these are the actual sequences. And this is how you might try to characterize what they're doing. So you will run ideally the same controlled experiments. You use the same instrument, same extraction kits, everything as standardized as possible, and you'll titrate in, different levels of virus, right? So this is what's shown here, where you have sort of standardized concentrations of virus from the easiest to detect. So this is, 15,000 copies to 15 copies, right? Probably per milliliter or something like that. So these should be the easiest to detect because there's the most RNA, and there's 1,000 times less RNA in those samples than there is. So you might imagine this is early in infection, this is sort of before symptoms, this is at the peak of symptoms, or this is a well-extracted sample, this is a poorly extracted sample, and so on. And the numbers here are these critical threshold cycle values. So the smaller it is, the sooner in the reaction do you see this characteristic transition from no light to light and rapid growth. And you usually want them all to be in the 20 to 30 range, because that's where the instrument performs most optimally. And you also expect, if probes perform similarly, you should have the CT values broadly comparable between, the different sets of primers and probes. And you can see that there are actually a fair bit of variation. So for example, while they're nucleoprotein, the vast majority of them is high concentration around 22, 23. If you go to a different probe that's targeting the RDRP gene, which is that slippage site, I believe, that's now up to 32. And it basically fails right here. If you go to a lower concentration, that's not sensitive enough to detect low concentration. So for example, you can already say that this is not as good a primer probe set as the others. And you would also expect, based on sort of very simple elementary college algebra, is that, there's 10 times fewer copies here. So, you need to do about 3 cycles of PCR. So 2 times 2 times 2 is 8, times 2 is 16. So somewhere between 3 and 4 cycles to cover for these, for this 10-fold dilution. So if you look across the columns, there should be about a 3 difference between all of these values. So, from 24 to 27, 27 to 30, 30 to 34, and that's about right. So, it has, you're in the linear part of the growth, so it doesn't go into, the suboptimal domains of RT-PCR reactions. However, this was in February, so the question is, a lot of the probes that are being used were set in February. So a reasonable question is that we have a lot more data now. The virus had 6 more months, or 7 more months now, to evolve as it spreads across the world. So is there any evidence that the viruses in the past 6 to 7 months have actually fixed mutations in those primer or probe regions? So we can now do a confirmatory study or a follow-up study to evaluate whether or not the probes that the CDC and other public health and universities and commercial entities selected early on in the epidemic, do they still work? And fundamentally, what we want to see is that you look at this probe and you look at the current distribution of viruses in the world, and you see, are there any mutations in those probes or primer regions? And if they do change, then test performance can degrade. So there's another tool that you can use. Again, you can click on the link at your own leisure and play with it. So this allows you to do, it allows you to put any range of coordinates that you want to investigate, and is going to show you two things. Going to show you among the 40,000 genomes that are currently available, high quality genomes that are currently available in GISAID. So let's look at the NCOV N1 forward primer from the CDC. So it's in these genomic coordinates, 28, 28.7, 28.306. And what this shows you here is a time series of how many sequences over time cumulatively have any mutations in this primer. And you can see that it's actually very, very few. So there's about 40,000 sequences total. And there's a slow and steady accumulation, but only about 75 sequences out of those have a mutation in that primer. So they could potentially not be efficiently detected by this test because the primer is now mismatched. And you can then look at the geographic distribution to see which part of the world these sequences are in. And most of these sequences are in the UK, and there are a few in the US and just a couple in Asia. So this is 1 of 3. You can look at the same in the reverse primer. So different set of coordinates. Again, only about 80 sequences that are different. Again, most of those are in the UK. And you can look at the probe. So the thing that actually are, where the fluorophores and the quenchers bind. And here you see a slightly different story where it's now 850 sequences that have some mutations in that region. So they could potentially not be detected. But they're also in different parts of the world. So they're in India, Australia, and some in the US. And you can try to do this for different probes and see how well they do, sort of comparative analysis. And in fact, there have been separate studies that looked at sort of laboratory measurements of what happens if you see that some viruses fix a mutation or carry a mutation in a primer or probe. So in particular, there's this one from the Journal of Clinical Microbiology in July that looked at a mutation in position 26340, and it was associated with failure of a QRT-PCR. I believe it's the Roche test. So you can see here, here's the table. So this is the COBUS test that has two diagnostic regions. So RDRP is fine. Again, these are all CT values, but note that all of these mutant strains that they put in had absolutely no readout in the E gene. A different test that uses a different probe had no issue picking up the infection in the subset of 4 individuals that they tested, but this fact is that they have zeros all the way throughout the column. The E gene means that half of the molecular target of this test has evolved enough that it no longer is recognized by the test. And finally, and I think this might actually be the most relevant part for you, is that there's been a lot of recent discussion in the press, which, if you remember my first lecture, is not necessarily something that you should take without a grain of salt. That a lot of the tests don't measure. They could be false positives or whatnot. The actual article that the people discuss, those posted in the New York Times, actually talks about antibody versus nucleic acid tests, and we're not going to do it, discuss it today. But what I do want to show you is some very basic understanding of diagnostic test performance. And each test, hopefully, if you didn't know anything about molecular tests, now you have enough understanding to stop and think that any of the number of steps there can fail. RNA can fail to amplify, probes or primers may fail to bind. Alternatively, you can have non-specific binding and a false positive. So the tests are not perfect. They will make errors. Hopefully not a whole lot of errors, but they will make some errors. So it is 100% true to say, without knowing anything else, that no test is perfect. And tests will make 2 types of errors. They'll make false positive errors, which means that if you're really healthy and you get a positive result, that's a false positive. The opposite of this is a false negative, when you have a person that really has the virus, but a test result is negative. There different types of errors, and depending on what is more important to you, false positives could be more important than false negatives. So for instance, one might argue that if you're really concerned early in the epidemic about doing contact tracing and potentially isolating people that might have been exposed, you might not care too much about false positives, but you'll care a lot about false negatives. On the other hand, false positives that require some significant treatment, for instance, if you get a false positive, you're put on a ventilator, which is a very, very invasive procedure. In that case, the cost of making a false positive mistake would be very high. So the cost of false positives, false negatives differs depending on the situation. But perhaps the most important observation is that when you have a large number of tests that are run in relatively rare conditions, the test can perform unexpectedly poorly. And I'll give you an example. So here's a screenshot that I took earlier today from what Temple reports for COVID-19 testing up to the end of August. So there's this cumulative testing, and according to this table, there was a total of 8,036 tests run, of which 368 returned a positive result. And this is about a 4.5% positivity rate. All right. So my question is, how certain can we be that these 368 are positives? And should we just trust these numbers as precise estimates of what's going on there? To help you understand this, there's another tool that you can play with, and it basically takes 4 important parameters for any test. How many tests you've run, so 8,063. The prevalence is something that we don't know directly. The prevalence is what is the true fraction of people that are infected with SARS-CoV-2 in the population. So we don't know this, but let's take it at 4.5% because that's what comes back at. And what Temple currently reports. And the other 2 parameters are sensitivity and specificity of the test, which are the 2 critical parameters of any diagnostic test. And sensitivity is defined as the proportion of positive cases that are correctly identified. And specificity is a proportion of negative cases that are correctly identified. And for the sake of argument, let's assume that this is a very well-performing test. So 99% sensitivity, 99% specificity. So let's run an experiment, which I did and took a screenshot of this. So, assuming that the true prevalence is 4.5%, if you do a random draw, that means that among 8,063 individuals, 381 were really infected. In this simulated experiment, there were 446 positive test results, of which 67 were false positives, and in addition, 2 infections were missed. You can sort of summarize the performance of the test by a couple of summary statistics. So there's one called accuracy, which is just the fraction of results, positive or negative, that were correct. So by accuracy, this test is great. But if you want to do sort of clinical decision making, or if you want to make a determination of how likely it is that a positive result is actually true, then what you should look at is a positive predictive value, which is the probability that a positive test really means that you're ill, or the negative predictive value, which means the probability that a negative test is really negative, so you're healthy. So this test will do very well if it gives you a negative result. So you can be quite confident that a negative result means you don't have SARS-CoV-2 or COVID. And the positive predictive value is 0.85, right? So it's actually significantly lower than either sensitivity or specificity. So there's a 1 in 5, or actually no, more like 1 in 6 chance that a positive test is actually a false positive, which isn't terrible, but it's not negligible, right? So for example, here you can expect that a fair number of tests were false positives. There's another awesome resource that is run by this FindDX organization. They summarize independent validations of commercial and other tests for SARS-CoV-2. And the independent part is important. So they actually have labs running control samples where from clinical data whether or not it's a positive or a negative case, and they evaluate how well the test is doing. So you can look up a specific test. So for instance, here's one, Perkin-Elmer SARS-CoV-2 real-time RT-PCR assay. And this was validated, by a Chinese hospital using a sample size of 242. And, they estimate that this is a highly specific test. So it doesn't give you a lot of false positives. But even then, because it's a sample size of 242, the estimate is like they didn't make any false positive discoveries, but we're not sure it's 100%. So it's somewhere between 98 and 100%. Right. So even, and this is one of the best tests. So 99% specificity is on the higher end of what the tests are doing. And here's the sensitivity, which means that, how likely the test is to detect a positive. So a lot of them will also be, near 100%. So here's, if you know which test you ran, I try to find which tests Temple actually uses, but it's not obvious to me. It's not anywhere where I could easily find it. Perhaps if you guys are curious, maybe somebody can dig into it and try to find which tests are actually run by Temple Student Health. The important parameters, it turns out, is the specificity. So if all you do is you keep everything else the same, so the number of tests that you've run, the prevalence of 0.45, sensitivity the same, but you change the specificity from 99% to 95%, which doesn't seem like that much of a drop. And in fact, most of the tests that you're likely to encounter in the marketplace will probably have specificity somewhere between 95 and 100%. This apparently minor drop dramatically changes what the test is doing. So now, again, it's just a computer simulation. So among 8,063 tested, 340 were truly infected. But now you have 717 positive test results, of which 382 were false positives, which means that if you get a positive result, it's only 50-50 that you really have it. And all you've done is change one parameter by a little bit. And the reason it is so sensitive is because we're talking about a rare condition. So 5% prevalence means that the majority of the tests are negative. So most of the tests that you run are in people that are not sick. And there's enough of them that come back as false positives that kind of skews your positive predictive value to the point where it becomes not as reliable as you might think it is. And for low prevalence, I mean, if you go to 90 specificity, now the positive predictive value is only 1 in 3. So a positive result is actually more likely to be wrong than it is to be right. Which is why it's very important when you look at a diagnostic test and you look at a specific population, you really need to know these sensitivity and specificity values. There are thankfully independent resources out there, like the ones that I linked, that allow you to get some of those estimates. Thankfully, if you do a large number of tests, it doesn't change the positive predictive value. You'll have a larger number of false positives, but the ratios are going to stay the same. So for example, if Temple ran 500,000 cases or tests, if we're able to test, I mean, Temple doesn't have 500,000 people to test, but say if we tested 500,000 people in the greater Philadelphia area using exactly the same test parameters, you would now expect 27,000 positive results and about 5,000 false positives, but the positive predictive value is still in the high 80s, in the 80 to 90% range. All right, so that's, do tests take asymptomatic carriers into account? You mean people that have the infection but are not, that are either early in the infection or don't have any symptoms? I mean, the tests don't really care. The only thing that is important is how much RNA do you have, right? So if the assumption is if you're asymptomatic, you have less virus, then the test is going to be less able to, detect that you have an infection. But they're tuned to be pretty sensitive. So if you go back to this slide here, right? So I mean, you're able to detect, pretty confidently over 3 orders of magnitude range of in viral load. So this would be a pretty dramatic difference between, so you might think that even if there's an asymptomatic person that only has 15 copies of the virus per milliliter of blood, all of these tests will still detect it if they're kind of on the tail end, but you should still be able to see it. So it really depends, again, on the test. Now, if you had an infection and you no longer have viral RNA in any appreciable capacity or quantity, then the nucleic acid test won't be able to tell you. But the test itself doesn't really care if you're symptomatic or asymptomatic. The key is how much virus you have.